Welcome to my channel World of Forensic. Today we are going to discuss about the examination procedures of diatoms. Introduction. As we have already learned in our previous video that diatoms are found in the water bodies. So in cases of drowning, there is a chance of getting this diatoms. So for the examination of these diatoms, prior to the analysis, the sample should be processed, must be thoroughly cleaned from the substances that like any type of pigment, other cells, mud, or any other substances that are present. And some of these methods which are followed for the process of extraction and examination of the atoms includes solvent 350 method, enzymatic method, colloidal silica gradient, centrifugation method, membrane filter method, dry ash method, nitric acid method, sulfuric acid method, and electron microscopy. Uh, to do all these, to perform these types of methods for diatoms examination, firstly we have to collect the specific types of bones like the sternum, or it can also be the body organs like liver, lungs, um, and these two are the main parts of the body which has the possibility of containing diatoms in case of drowning cases. Solvent 350 method. It is the fastest and less expensive method and is most performed in case of freshwater cases. Here the sample is mixed with formalin, then rinsed and centrifuged 3 times using distilled water. After centrifugation, the pellets are removed from the test tube and added with 8 ml of solvent 350. Then the solution is incubated for 2 hours at 50 degrees. After that, it is centrifuged again, and after centrifugation, we get the supernatant and the sediment. The supernatant is thrown away, and the sediment is mounted on a slide and seen under the microscope for that. The enzymatic method. Enzymatic methods are basically used for uh, tissue samples. Here, the concentrated nitric acid and proteinase K or peroxide is added to the sample and kept for 12 hours. Then the sample, after 12 hours, they are centrifuged 2 times. After centrifugation, it is rinsed with distilled water, and after rinsing, this is mounted on the slide with nephrax and are viewed under the light microscope for observing data. Colloidal silica gradient centrifugation method. This method is applied if the tissue samples are from liver or lung. The tissue layer is added to the saline solution and centrifuged at 12 degrees Celsius. Then distilled water is added to it. After that, it is taken and mounted on a slide to view under the microscope. Membrane filter method. It is used in presence of either damaged atoms or presence of any inorganic materials. Here, the blood of the victim is taken for the analysis. The blood is taken and filtered through the sodium didecyl sulfate and nitrocellulose membrane, which have decreasing size of pore. The membranes that were used for filtering are then dried and again filtered using distilled water. Then these membranes are examined under microscope. Dry ash method. So this method is used in case of bone marrow sample. So when a bone marrow sample is used for the examination of diatoms, the driest method is used. Here the let's take 5 gram of bone marrow into nitric acid and then it is burned in the furnace with the ash. Then with the release of the ash, it also releases the diatoms. Then these diatoms are analyzed, and this bone marrow sample helps to identify whether the body was drowned before or after death. As the diatoms, the highest level can reach the bone marrow of a person. Nitric acid method. To perform the nitric acid method, we should first have a small section of the sternum bone or the femoral bone from where we can get the diatoms. And then acid digestion is carried out by treating that part of the bone with concentrated nitric acid. Heating is done until clear solution is obtained from the bone and the nitric acid. Then hydrogen peroxide is added to that clear solution and again boiled. After boiling for some time, when it is cooled and then it is centrifuged. By centrifugation, we get the supernatant and the sediment. The supernatant is decanted off from the centrifugation and the sediment which is obtained as observed under the microscope. After observing if diatoms are present, then we will get to see these diatoms under the microscope. Sulfuric acid method, it is also acid digestion method. Here the calcareous compounds are removed from the sample so that they do not form the gypsum crystals. So after settlement of the sample, the supernatant is discarded. Then concentrated sulfuric acid is added to the sample until it becomes twice the original volume. Now enough potassium bicarbonate is added for a saturated solution to make it a saturated solution. Then it is left to stand for 24 hours or to speed up the reaction it can be put in water bath at 60 degrees Celsius. Uh, After putting it in water bath we get a gray sediment and the test tube in the sample. Then, after getting the gray sediment, the supernatant from the test tubes are discarded and the sediment is rinsed several times before viewing it. In electron microscopy. The electron microscope or the dark phase microscope is used for the analysis of the diatoms. To examine the morphology of the diatoms, two types of electron microscopes can be used. One is the SEM, that is scanning electron microscope. The other is the TEM, that is transmission electron microscope. The SEM is used to view the gross morphology of the diatoms, all the fistulas, both internal and external parts of the diatoms. And the TEM is used to see the delicate details of the diatom fistulas. Thank you for watching my video. Please subscribe to my channel and press the bell icon for notifications of my videos.